Okay, welcome everybody. This is our figure making workshop. So the objectives are first to practice using PowerPoint to make line art. If you've used Photoshop or you already use PowerPoint, this might be review for you, but if not, take notes, practice. I'll go and actually do some of the exercises for you. You're going to learn about the very important points of handling images. Ryan is going to lead that section, and it's fantastic. I learned things from him I never knew, which I wish I had known when I did my thesis. And Tom is going to take you through taking your numerical data and actually making graphs. Graphs that are legible, graphs that people want to look at. Okay? So, moving images, I don't think we actually will be doing this. This is a little bit. You're going to need some of that. So moving images and from one application to another. Now, this is an important reminder I had from before. So be sure that all your pages have 1.5-inch margins. Is that still true?
Yeah, we actually are changing that this year.
I told you to make any announcements.
I'm sorry, I didn't know that was on there.
That was on there from last year. Okay, a few frequently asked questions about figures. I'm going to let you guys weigh in on what you think the answers are. All right, do my figures need to be inserted into the text? What do you think? I hear no. Lots of no's here. Come on, get in. No, lots of no's. Okay, so they don't need to be. All right, you can do it any way you want. I would highly recommend that you don't insert them into the text unless you're absolutely sure the text is done and you're not going to change it. And then you want to go in and insert because you think it looks nicer, fine. But it is a pain because when you insert text and then you insert figures and then start moving the text around, it just messes everything up. So there are two good options for you, and that is have full pages with figures that are intercalated into the text, or have an appendix at the back that has all of your figures. And the concentration advisors can weigh in on what they prefer. Yes?
For HDRB, I kind of prefer it to be interpolated into the text.
Me too.
Ditto?
For Neuro, you can do what you want. Seems like the easiest for you would be to just give each figure a separate page and intercalate it into your thesis. But do as you like.
All right.
You want for us, for me too.
All right, question number 2. Can I use published figure? Can I use a published figure in my thesis? What do you think? Yes, and I heard somebody say
If you cite it.
if you cite it. So if yes, and if you cite it. So you need to cite it in the figure legend, you need to cite it in the text, and you need to add that citation at the back in your bibliography. Okay? What about if you don't exactly use a published figure, but you base your figure on a published figure? So you basically get the idea and the inspiration, you make a figure that's completely laid out exactly like a published figure. Can you do that? Do you still have to cite? Yes. Okay, so modified or based on a figure from this reference. Okay, so make sure you pay attention to those details. All right, and that is, if I find, I forgot I had this one in here. If I find a figure that contains information I need, but also has many additional graphics, can I just white it out? Can I just white out the extra stuff? Can I block it? What do you think? Is it, can you do it? Yeah. Should you do it? It will look bad, and it will look very obvious that that's what you did. Okay, so although we're not expecting you to be professional illustrators, we are expecting you to put some time into how you represent your information graphically. Okay, so don't do that. Do my figures need to be in color? How do you guys feel about color? Like it? Yeah, we all like color. Sometimes we like color a little too much, right? So do they need to be in color? No. Do people like to use color? Too much. Okay, and we'll talk about how to use appropriate amounts and numbers of colors. Do I need to represent every molecule in the pathway or process I'm studying? It's biology. It's complicated. So actually, you might want to have a figure, and we'll go over this, like a figure that's an overview, and then maybe box in the areas that you're specifically going to focus on and make a new figure for that, right? You might want to visually give us a context, but not give us every single bit of detail unless it's actually pertinent to what you're doing, okay? You guys jump in whenever you want to add something. So if not, how do I know what the correct amount of information is? This is a question we get a lot from students. So how much detail is too much detail? You guys want to talk about that, or? That's something you should actually talk to your PI about, okay? So if you feel like your figure is getting cluttered, then go to your PI or go to the graduate student or postdoc that you work with and talk about, okay, how do I clean this up? How do I have the integrity of the information, but yet not have a cluttered figure, okay? And sometimes that will actually be a stepwise process where you have two different figures or two panels within a figure. Okay, so we're just going to go through a few figures that I found by Googling. All right, and let me know what you think about these. Okay, so here's a figure from Kaijin. Impressions? Do you like it, dislike it? If yes, why? If not, why not? Take your time, look it over.
There seems to be too much going on.
So too much going on here, so specifically because of all the arrows, maybe? Or just colors, lots of proteins, every single possible protein that comes out of the Rho pathway. Probably you're not going to be covering every protein that is signaling downstream of the Rho pathway, right? What else? Let's talk about the use of color. So there's a lot of colors. How might you use color differently for this figure? Yeah.
What are the pathways that you're interested in? Make everything else black and white.
That's a good one. Maybe not black and white, but a grayscale, right? So use color to emphasize what's important for you, right? So what if you have this, not have every single pathway, but there might be a few parallel pathways that are relevant, but not critical, right? So if you have most of these in grayscale and then pop yours out with color, that's a really good way to more clearly communicate what's interesting, okay, and what's important. For example, I think, why waste this nice bright blue on arrows? Why? Right? It's a great color. Use it for something that's important. So use black or gray for arrows, okay? Because it's not informative other than directionality, right, and interaction. Make sense? So we're not expecting any of you to be graphic designers. Some of you might have a really good art background or graphics background, but many of you won't. And we're not expecting you to be experts at this. But go to some journals that have good figures that you think are clear and crisp, and sort of model your style after that, and that will help. Let's look at a few more. Yeah?
I want to come back to this.
Where?
Another thing to be aware too, if you look at those right under row A, they have that dark blue black lettering.
Oh, this.
It's very difficult to see that lettering with that color in the background. So also be cognizant for your reader's sake to be able to have labels if you put them on there to be more clearly visible.
Okay, so that's another good thing. Make sure there's contrast. If you're using a dark color, don't put a text note right on top of it. And I'm repeating this because we're being taped and people not here will need to know what Bill just said, okay? So I will repeat all questions for our videotapers, tape people in the audience. All right, here's another one. What do you think of this? There's some really good ones. You go online, you find some figures that are just atrocious. What do you guys think of this one? Ugly. Ugly, right? Isn't that your gut feeling is when you see this, like, ugh, puke yellow for this letter. It's like, why? Why? It's just not necessary. First of all, they're just, names of proteins. You don't need to really make them that color. And again, there's no way to know which information is the most important here. Okay, so there's no way to distinguish what I should be looking at. And I don't think any single senior thesis would deal with this number of pathways. There's no PhD thesis that would deal with this number of pathways, as far as I know. Okay? All right. What about this one? This is my favorite. What do you guys think of this? What are they even trying to show me? You. What do you see? When I first looked at this, I almost thought they were trying to indicate that they were zooming out, right, from this. But then I'm not sure, because these are labeled as microtubules, so maybe this is actually just one specific area along a microtubule. I don't know. Microtubules, are they shaped like this? Flat? not as far as I know, we actually have electron micrographs that show that they're not flat, so I'm not sure what's going on here, but this is actually a misrepresentation of scientific knowledge, right? And this blob is like mitochondria, and there's only 2 in this cell. Okay, so again, what you're showing should represent what you need to communicate visually, right? No visual will stand on its own. The figure legend should complement the synthesis of the 2 with the information, should communicate clearly what you're trying to tell us with this figure. But this, generally don't use 3D effects and shadows that actually occlude information. There's 2 reasons. Not only does it obscure information, right, but it actually makes your file size a lot bigger also. When you don't need shadows, you don't need these 3D effects, and you don't need gradients, don't use them, okay? Because even with vector art, when you start to accumulate all those things, it makes the file size enormous, okay? All right, anyway, that was a treat. This one is not so bad. It's maybe got a lot of information, but you can imagine taking something like this, black and white and gray, right, and then adding in a piece that is important to you. What if you only care about focal adhesion kinase, right? You could pop that out. Not so bad, right? Might be a little busy with respect to the text, and potentially you could put a legend that tells us what these things are, but overall, not as bad. This is my last one, and this one, I like the idea that they're showing you that there's a legend, right? But again, I think that they're showing us a lot of little bits, and maybe this might be an overview, right, of a focal adhesion or what's going on here, but this might not be exactly what you're going to focus on. Okay? Any feedback? What do you guys think? You guys will be interactive because I won't go forward in future slides until we hear from you. Okay? Anybody want to say anything? Anyone have any experience with horrendous figures that you couldn't understand what they were saying? Is everyone unanimously going to make sure that your reader does not feel like that when they look at your figures? That's really the point. That's really the point. When you see a figure and you just go, oh my God, who put that together? You want to make sure that when the person reading your thesis to grade your thesis doesn't make that face and think that when they're looking at your figures. So put yourself in their place and go, okay, I'm going to clean this up and I'm going to make sure it's clear. All right? All right, moving on. So what if you are a student working on cell motility and you want to focus on rearrangement of the actin cytoskeleton and adhesion to the extracellular matrix? So I'm just making this up. But if this was from a figure in a published article, but it has too much information, okay, maybe you will redesign it, right? But a figure like this might be useful to say, okay, this is the big picture of my problem, cell motility. These are the players involved. And then I'm going to specifically draw a more detailed figure at what's going on here at the leading edge of the cell and what's going on here when the cell is forming focal adhesions, right? So you can imagine a 3 or 4 panel figure in which you have the big biological picture and then more details about what your thesis is specifically about. Does that make sense? Okay. Now, the one thing that's a little bit difficult is it's really easy to draw circles and squares, right? Simple, right? But if you're drawing a cell, and pretty much everything we deal with in biology is irregular in shape, right? So how do you get this? I literally put this together in 5 minutes. Okay? It's 3 circles that I stretched out and morphed a little bit and played with their colors. Okay? So this is actually not so difficult. And that's what we're going to cover right now, is how to draw irregular shapes in PowerPoint. Sound like fun? All right. Okay, so let's first look at the toolbars. I confess I have an old version of PowerPoint, but the toolbars generally stay in the same place and generally always look the same. All right? So when you pull up your palette, if you go to View and you go to Formatting Palette, depending on which of these up top you click on, you will see either the objects or you will see these different options. Okay? When you're dealing with objects that you're playing with, you're going to specifically care about size, rotation, and orienting, as well as color, weights, and fill. Okay? Those are the 2 aspects of the palette that you care about the most when making figures. Now, this is fun. Who has experience drawing with PowerPoint? Do you like the default that Microsoft comes up with, like the blue line with the shadow? You don't love it? Okay, so you know how to get rid of it? Does everybody know how to get rid of it? So you can choose a default. You can actually say, okay, I don't, every single time I open a file, I don't want to see that blue color and shadow. Okay, I want it to look like what I want it to look like. So you can actually go to the Format menu and go down to Shape, and then Control click, and you will see Set as Default Shape. So if I was to do this, I'm just going to get out of the presentation mode for a second. If I click on this, Control click, right, I don't want to set on default yet. So let me go to Format, Shape. Let me change this so it's not so nasty looking. And let's just make it gray. All right, now I'm going to Control click on this and set this as my default shape. And now if I go to this palette, this is another neat trick that I actually just discovered today. You don't actually have to draw. You can just click here and you get a new circle, which is nice. And they can, well, I didn't click a circle, but you get the picture. Okay, you don't have to have that silly blue default. Okay, get rid of it immediately. Now everything I make from here will be exactly this shape. All right, so now in terms of getting it to be a freeform, like we said, most of the figures you're going to work with are not regular shapes. They're not going to stay as circles and squares and rectangles, right? So how do you actually deal with that? Okay, so again, you're going to just click on, if you guys have your computers, you can actually practice this and just take a look at the menus, make sure you can find them. So you're going to click on it again, and you're going to actually say, convert to freeform, okay? So if we do this, okay, Control click, you can see right here, convert to freeform, but it doesn't look like it changes anything, right? It looks the same. So in fact, there's a second step. So you now have to not just convert to freeform, okay, but you actually have to double click it again, convert to freeform, and if you click it, Control click again and say edit points, now you can see the change in the object. Do you see that? It changes to a red outline with these editable points. Now, with editable points, you can basically do anything you like. You can grab, actually I'm going to cover the next couple slides just here. You can grab the path, okay, and pull it out. You get these whiskers. You can pull them up, make them longer, move them around. Okay, and this is how I made that cell in 5 minutes. I decided on the shape I wanted, okay, and I played around with it. What else can you do? You can also go right over a point and Control click, and you can add points and you can delete points on that path, or you can open the path entirely. Now I've opened it, I can actually move this out separately. Well, I'm having a problem grabbing that point, but you get the picture. There it is. Okay, so you can open it and then you can join it with another object. So what's the advantage of joining it with another object? You guys ever tried to do that where you group two things and then you try to make a gradient and then the gradient actually shows the two original objects? So it's not truly a single object. So again, this is something that I don't have in my version, but that Tom pointed out to me, that a student pointed out to him, that you, in the newer versions, you can do this. So I'll make sure that Lisa posts this PowerPoint, but if you go to this URL in, I think it's Microsoft 2010 and newer, 2011, you can actually fuse objects. So if you want to make a special object that actually, like ATP synthase, for example, this is my favorite example. You always see ATP synthase represented as a rectangle and a circle, right? Like this mushroom thing. You have something like that where you actually have a transmembrane protein that has a cylinder, but you want it to be a single object in which the extracellular domain and the intracellular domain have different shapes, and you want to fuse them, you can do that in the newer versions of PowerPoint. So I don't have a lot more to say. Is there anything I missed? There's a few other things that if you guys don't know about that are very handy. When you go to this alignment, if you're making something that has, like, say, neurotransmitters at a synaptic junction, and you want to make a lot of them, and you want them to align along the membrane, you want them to be spread out, right? You make a whole bunch of them, and you can use the align and distribution functions to make something like that. And I can show you quickly if you want to see that. For example, let's say these are vesicles. Look at that. And so it's easy to do this, but let's say I want them spread out. I can select them all, and then I can go to the alignment and say, first of all, I want to distribute them vertically. Okay. And then I also want to distribute them horizontally. Okay, so you can set the horizontal and vertical margins. So right now it's set to the margins of the slide, and that's why it's going vertically or horizontally across the entire slide. If you unclick that and you decide, and where you start with each object, those will be the limits of the group. Okay? It's just that currently on my slide, it sets the margins of the slide. But there's a really quick way to just copy a whole bunch of little things, like vesicles, and then distribute them the way you want it. Does that make sense? I hope these were useful tricks. I'm done in 30 minutes. Is that good? Do you guys have questions for me? You guys are quiet. Did you not get your coffee on your way here? There's coffee here. I see coffee. All right. So actually, I would say the gem of the show is really Ryan's presentation anyway, and it's very important that you pay attention to this and learn how to deal with images, and do not crash your PI's inbox with enormous files. Do you have an adapter? Yeah. Okay.
So, do you guys all use PowerPoint? You have it, a copy for yourself, or do you use anyone use another program? Nope. Yeah? What's it called?
ChemDraw, like BioDraw together.
ChemDraw, okay. But for this purpose, making figures, you'd probably be using PowerPoint?
For like, because ChemDraw comes with BioDraw, which has like little shapes and has like a lot of biological shapes, and you can make figures. Is that the BioChemDraw? Yes, yes.
Any Illustrator users?
Yeah.
How are you? Powerful.
I love Illustrator, it's my favorite.
I hate Illustrator.
I love Illustrator.
It's too hard to use, right? There's nothing you cannot do with Illustrator, but it's going to take you a PhD to learn how to use it. So like Cheryl showed, I feel like PowerPoint has really come into its own as a really powerful way to make figures. And it is indeed how most scientists now make figures for their papers. I need the mic, sorry.
You can use it with
Turn this on, I guess. Is it on? Is anyone using PowerPoint version 2007 or 2008? Or do you all have 2010, 2011? Some people still on 2007, 2008? All right, so they keep changing the program a lot. And that was a big change, the version 2007 to 2008 to 2010, 2011. I tried as much as possible to include all the programs, Mac and Microsoft, for all 4 years in the instruction sheet that I handed out. So I hope that's helpful. But if you guys ever have any questions about any of this stuff, just email me, because I thought about it a lot, and I should be able to help you relatively quickly. So my part of the talk is really about rendering your images and rendering them with the same quality and same attention that we do whenever we produce figures for scientific research articles. And actually, after this talk, you may notice that there are some scientific figures that are not so good. And here, with just a few tips and tricks, I feel like you'll be able to produce images of the highest quality. Right? And it doesn't take that long to do, and there isn't that much to know. But there is, I don't know if it's correlation or causation, but there is a relationship between theses that win prizes, Summa and Hoops, and the quality of the figures, right? So I think if you want to be in contention for those awards, or you want to impress people who read your work, good figures are really important. Okay? These are some figures I like. I did my PhD in the Lichtman lab where Ian Boothby works, and we do a lot of image processing. So I learned all this through my time in the lab. Otherwise, I'm the neurobiology academic advisor. So I've tried to distill this down and share with you what I know. And there's some resources you can find at this link. Basically, it includes the PDF of this presentation and the instructions I handed out, as well as some of the sample images that I'll work with very quickly in this presentation. Okay, good. So what I have noticed looking at theses and maybe the mistakes I've made myself is that there's maybe 4 major categories of errors and mistakes that figures have which you can easily improve, right? One is when your figure looks like this. What's wrong with this figure? It's too dim, right? And who knows, but it looks like there's a lot of empty space in the figure. Why include that? It's not necessary, right? You really want to highlight the information that's important and not include distracting or irrelevant information in your figure. Right? So this one's, the images are too damn dim and they contain too much empty space. And there's a simple solution for this, which is adjusting the levels. And you guys have access to powerful image editing software. Harvard no longer gives students licenses to Photoshop or Illustrator, in case you haven't noticed. But that's a blessing because there are simpler programs with all the same features that you can download for free. So we'll talk about them. And after you adjust the intensity values and crop your image, you can get something that's much better. You have to remember who is reading your thesis, right? Who's reading it? A bunch of old, curmudgeon-y professors with bad eyesight, and they're barely going to be able to see this. So you want to make sure that you're not giving them a headache with bad figures, right? Yeah. Oh. To take it down a notch. Okay. So the other thing that we do wrong is when we adapt figures from PDFs, right? Sometimes the figure looked good before we took a screen cap and put it in our presentation, and then it looks all pixelated, right? So we'll talk about how you can easily avoid that, and it's simple, so that your figures look as good as they did when they were first published. Okay, a related issue is that the graphs, the figures, maybe in BioDraw or whatever that program is, or MATLAB, the schematics that you make end up looking lousy. Again, pixelated and not properly done. And there's no reason why these images should look pixelated. Really, they should look nice and crisp so that you don't see any pixels at all, no matter what program you use. So we'll show you quickly how to do that. And the last thing, the last major problem is people who turn in theses that are so big, they don't even fit in my Google Drive or my 20 gigabyte Dropbox. It's a problem. So you don't want to be one of those people. There's absolutely no reason for it. And ultimately, even with 10 pages of full figures, there's no reason why your thesis need to be more than 20 megabytes. For most of you, your thesis will be substantially less than that. So we'll keep those things in mind as we go through and try to understand what figures are and how we can make them, professional quality. So what kind of file formats do you guys think of when you think of figures? TIFF, what else? BMP, did I hear a BMP out there? Oh, PNG, okay, good. Anything else? JPEG, okay. So these actually are a very specific class of figure and generally not the file format that your figures will have. Okay, when we are making figures, we're going to be talking about using file formats like PPT because try to convince you to make them in PowerPoint, but ultimately you save those figures as PDFs. Why? So we're going to look into that right now. So if I open a PDF, one that I have on my desktop, it's called Advising Matters 101. Here we see what ultimately is a figure. It's information that is being presented to you. It's presented in a PDF format. Why? Does anyone know what PDFs allow us to do in terms of visual information? Probably not, right? What PDFs can do is they can include both raster images and so-called vector images, right? And those two types of images are the reason why, if I zoom up on this PDF and the photo of David Haig or the text, you're going to see something very different. So what happens in a PDF when you zoom up on the text, like really high, like 1600%? Does the text look pixelated? No, it just looks perfect. It's like, let's try to zoom in. So as we zoom in, right, the text looks the same. It never gets pixelated magically. It's always looking good. The edges are always sharp, even at 6,400 times zoom, right? That's amazing, right? The reason for that is because the text and other objects in here are what's called vector images, meaning they're mathematical expressions. They're not made of pixels. So we'll look at that. What about David Haig? What's going to happen if I zoom up on David Haig, like they do in Miami CSI? Just zoom, zoom, zoom. We see what he's eating, or maybe what he wrote on his little notepad here. Student is boring, don't accept it, OME. What happens if I zoom up on David Haig? He's going to look really pixelated, right? We're not even going to be able to see that it's a human being if we zoom up to 6,400 times, because this image was taken with a certain resolution, which is displayed in pixels. And when you zoom up, yeah, what color eye? Is that even an eye? That's something that belongs to David Haig. But the amount of information that's there is not sufficient for us to see the level of detail we're looking for here. So that's kind of the difference in the two classes of objects that you will have in your figures. Almost every figure is going to have a raster object and a vector object, and you want to be able to display both of them correctly. And that's the reason why we end up using PDFs and PPT to make our figures. So going back to the presentation, just to kind of recap what I said. Let's see here. Figures are a mix of raster and vector objects. Raster objects are things that you're going to be getting in the lab, probably, like scans, if you were to take a scan of something. Microscope pictures, photos, screen captures, all that stuff is pixel-based. These are the type of things that have extensions like TIFF, PMG, JPEG, BMP. These are the kind of images we associate with figures. Pixel-based, set number of pixels. When you take the image, if you zoom in, you see pixels, right? You can edit these in Photoshop if you have access to it, and I believe you do, because I think it's on the computer lab computers. But if you want to work on your own computer, there's a freeware program called GIMP for both PC and Mac. And I don't know why it's called GIMP. It's not because it's gimpy. It's, I think, graphic, something, I don't know. Your guess is as good as mine. But it's a great program, and it's free, and it does everything Photoshop does as far as we need. Vector objects are things like shapes that you draw in PowerPoint, text, Excel and MATLAB graphs, and probably any other type of figure you export from some other program you're using to display and collect data. And these guys are not pixel-based. They're just mathematical expressions that are redrawn at every zoom. So you can zoom in without blurring infinitely. And you can create and edit these objects in PowerPoint or Illustrator. We recommend PowerPoint. PowerPoint and PDFs can handle both vector and raster objects, which is why it's so great to use it whenever you're creating your figures. Photoshop only handles raster objects. Any vector object you put in Photoshop will be rasterized, which means it will make it into pixels at whatever resolution you put it in there. So working in PowerPoint, that never happens. You can actually keep vector images vectors, so the text never looks pixelated. Okay, a little word on file formats. TIFF and PNG are generally the preferred file format whenever we're keeping data around, reference data, because they use a lossless compression, which means they can actually shrink the size of the file, so compress it, but when they do it, they can retrieve all the information that was originally there. And PNG compresses more, so it's a smaller file size, but either of these are going to be fine, more or less. Generally recommend keeping your original file as a TIFF, and as you edit and modify it, save it as a PNG. They're a little bit smaller, so if you have lots of copies, it just takes up less space and just as good. JPEG, on the other hand, is often the default file format that saves from some of our microscopes or other programs, generally it's not as good because it uses a lossy compression, which means it actually throws away data it thinks is of marginal importance whenever it stores your image so that that image can be smaller. Okay, but for us, people who are in science and care deeply about the information in their images, we're going to steer clear of that until the very end. Eventually, we use it because it compresses the photos a lot, but as long as we use it at the very end, probably we won't notice the difference if our last picture is a JPEG. So the PDF automatically converts your images to JPEG by default. That's fine, the last step. This is an example of an original PNG with the file size associated with it. High quality JPEG is there. You can't really see the difference between them. Low quality JPEG, much smaller. I don't know if you can see it, but you actually start to see some of these artifacts of compression in the image itself. That's what we want to avoid. And if you're opening, modifying, and saving the JPEG many, many times, you start to produce some of these artifacts. It starts to look crummy. So you might as well just use the PNGs and not worry about it. OK. So the raster image, as we said, is just a grid of pixels. And each pixel is a block with a grayscale value, from black to white. For an 8-bit image, you guys have heard of 8-bit images before? What other kind of bit images do you run into? Do you use different, on your programs or digital cameras, always 8-bit? 12-bit, 16-bit, right? So what does the 8-bit mean? What is a bit? Some kind of digital stuff, right? Digital, what does that mean? It's either on or off, right? So on is one state, off is another state, so there's two states. And we have 2 to the 8th power whenever it's an 8-bit image. And what is 2 to the 8th power? It's 255, which in this context just means our image is going to be, we're going to take that whole color scale, not color, but grayscale from black to white and chunk it up into 255 bins. And a pixel can have any of the values of those bins. So just for the purpose of quantification, if we look at a part of the image here, you see those pixels, and each pixel has a value which is quantifiable. If it's perfectly black, it's 0. If it's perfectly white, it's 255. That's how an 8-bit image is constructed. 12-bit image, what's 2 to the 12th? It's 4,096, right? So we have a much larger number of discriminable bins that we can use to populate our photo. So it actually contains more information. Problem is the human eye can't see all those 12 bits differences. Computers can. So we often take images that are 12 bits to capture more information, and we can quantify them because the information's there even if we can't see them. But for display purpose, normally we just use an 8-bit image because it's much lighter file, much less information. Okay, good. So the other nice thing about having an image that's made of information is that we can look at a histogram to adjust the values so that we display it in the best possible way for the human eye. So what we have and what we can find in any of these imaging programs is a simple histogram that shows the number of pixels in your image with a certain value over the different values that the picture contains. So how many pixels are pure black versus how many pixels are pure white, and every discriminable grayscale value in between. So we have an image like this. We can see what pixel values it has. In our image, what is this image going to look like based on the histogram you see? Dark, right? It's going to be dim. We don't want to show a dim image, do we? Do we? If the image is dim, maybe that's what we want to show, right? Right, we have to be careful here. So here we're walking into some quicksand. So in the imaging lab that I worked in, basically what we had as a quick and dirty rule was whenever you're quantifying an image, you have to quantify the original image before you modify how it's displayed. But if you want to show it to someone and show it to someone with human eyes, you want to show the information that's in there in the best way you can. And that usually means adjusting the levels so that people can see the differences in the information that are there. The part that gets tricky is when you're comparing one image to another, because you can skew one to make it look bright or skew one to make it look dim, and that's obviously going to misrepresent the data. So whenever you have two images that you want people to compare, you have to adjust them the same way. So you put them in the same image, and then you adjust the whole image. But for a single image, it's OK, for the sake of display purposes, to modify it to really bring out the information that's in there. So we can adjust the pixels to help the viewer see the information. And what we end up doing is stretching the pixels out over the entire display range so that the eye can see the differences that are in there. So an image like this block of pixels, after we adjust it, is going to look like this, where it's easier now to see the differences between these pixels and these ones that are on the fringes or margins here, right? And the way in which we do this, it's the same in Photoshop or GIMP. And there's instructions that I handed out which go through this, I hope, in a comprehensive and intelligible way. It will allow you to do this if you forget what I'm saying now. This is how the actual histogram will look, because we have a lot more pixels in our images generally. And what we have are three different triangles at the bottom of the histogram, right? The axes aren't labeled, but this is 0 to 255, the number of pixels in each bin. Here we have 1, 2, 3 triangles, one black, one white, and one gray. The way in which we make our images beautiful and really pop out is first to move that triangle at the 0 value a little bit to the right. So it's setting 0 to the first value where you actually find pixels. So in this image, there's no pixels that are at 0. So we take our value of 0 and move it up to this pixel. So now we've set the background of our image to pure black. Then we take the top triangle and we do the same thing. We move it down, this time to the left, until it hits the first pixels that are actually displayed in our image. So those pixels are pure white, right? So that's a pretty simple kind of adjustment. It generally helps out, and it just means that we maximize the differences between the range of pixels we actually have. But the more interesting adjustment that we can do, these are just some tips look at later, is to use this third triangle, which is called gamma, right? Does anybody know what the gamma does or heard that name before? I know Ian has. Ian, what does gamma do? Putting you on the spot.
So usually, like, in the range that you specify, there's like a linear change in intensity. So, like, if it was like 0 to 100, then 50 would be exactly half. But with gamma, you can make it varying degrees of nonlinear, so you can like bring out the middle values or bring them down.
Right, without sacrificing the values at the edges. So let's take a look at what Ian just said. It's all 100% correct. When we move this triangle, we're bringing out the midtones. So those values in our image that are a little bit dim, we want people to see them. But if we just moved all the highest values forward, so then these pixels would all become white before we could really start to see these clearly. And we don't want to lose the information that's here, saturating them by making them all the same value. So we have to be careful how we set the edges, but then we want to give these values a boost, because generally it's hard to see values that are only at 120 on the grayscale level. And the way in which we do that is the third triangle. And when we third triangle, we have our old pixel values, we have our new ones. What it does is puts a non-linearity between the mapping of the old and the new, such that the old value here in the middle was 125, would have been 125, but now 125 is much brighter, even though we didn't do a lot to the upper and bottom pixels. So that just boosts up the midtones. It's really helpful for making your images look good. And just to show you how quick and easy that is, I have this image in GIMP, which already looks pretty nice, I have to say. I took it. I did a good job here. But we can make it a lot better, right? And these are in your instructions, but basically there's just a menu item that you go to. You see these histograms, just like I showed you before. When we have an RGB image, we do each channel separately because they tend to have different pixel values, different histograms. and then we move our white one up, we see that it becomes brighter. The red becomes brighter. We get rid of some of the background because it's a little red, and we don't need that. And then we boost up our gamma if we need to bring out those midtones. Not too much. Don't want to screw up the background. And you just play with them until it looks good. And then if I go to green, do the same thing. We see that in green, most of our pixels are clustered down here. We can look at the histogram with a logarithmic scale that shows in a little bit more detail those pixels at the highest levels. In our case, we've already got a lot of pixels at the highest level. We don't have a lot of background. It's already set to 0. So we just kind of adjust the gamma a little bit, and we can bring out those green midtones that were otherwise lost. We do the same thing with blue. We move it forward. A couple pixels will be saturated here. Generally, they're kind of artifacts in the image anyway. The baseline background looks good, and then we boost up the blue a little bit until our colors look the way we want them to. And now it looks better. And the way you can tell it looks better is if you undo the levels, and you see how much it pops out whenever you adjust those colors, right? It's a big difference when you have a color image. And it's important to really show the information that's in your image. And it, like I showed, takes a minute or two. OK, so that alone is going to do a lot to make your images look good. Cropping, there are instructions in there too. That's even easier, just one button. Now, there's a big question that comes up, which is what resolution do I need in my raster images for my figures? So what resolution do you guys think you need? Yes, the answer's on your sheet, but think about it. What's the resolution of my screen? Does anyone know that? A projector or the screen you're looking at on your computer? How many pixels per inch does your display have? No one knows? Not too important in your lives until this moment. Now it is. 72 pixels per inch for your display. 72 pixels per inch for the projector. When you print something, however, if you print at 72 pixels per inch, it won't look very good. So generally, for printing, we print at about 200 dots per inch, or pixels per inch. 220 is what I recommend for you. Okay? So we want our images to look good on paper because our readers, some of them at least, are going to actually read the hard copy of your thesis. Some of them will look at it on the screen, and for them it doesn't matter, as long as it's 72 pixels per inch. But for those old-timers who like to read paper, we need to make sure that our figures look good when they're printed out. 220 dpi. The screen only shows 72 dpi, so you can't really tell the difference between a 72 dpi and a 220 dpi or ppi on this screen. So this is the reason why when you screen capture a figure from a paper, it can look bad whenever you end up showing it or printing it out. Because the screen capture you takes at the native resolution of your display, which is 72 pixels per inch. And if you try to show that or print that, it's going to be much lower resolution than the original, probably. So the simple workaround for that, which hopefully you guys already know if you've been doing scientific presentations, is to take your original image and blow it up to like 300. 300 is fine, 400 if that's your option, percentage, so that now it's 72 pixels per inch, but it's 300% of what you're going to need in the end. So then you take your screen capture at this relatively relatively large in size image that's 72 pixels per inch, and then when you put it in PowerPoint, you just drag the corner and shrink it down. And then by shrinking it to the size you need, it keeps all those pixels and it compresses them so that ultimately it's a 220 pixel per inch image. Does that make sense? OK, so that's critical if you're not already doing it.
Brian, is taking a screenshot the same as copying and pasting from a PDF?
Yeah, I believe so. I'm not sure about that, though. I know that if you have an image open and you try to select it, copy, paste, and put it into a different program like Word, it will do it at a lower resolution than the resolution of your screen. So generally, when you put images in different programs, insert them or drag them from the file. Don't copy, paste. So what resolution? We've talked about that. How do you get these 220 pixel per inch images? It's really simple. And just to go through this conceptually, let's say your image is 1 inch by 1 inch, and it's 10 pixels per inch. It's a raster image. How many pixels is that total? 10 pixels per inch by 1 inch times 10 pixels per inch by 1 inch. It's 100 pixels, I think. Mental math, so you can double check me. Now, what if you wanted to make this picture 100 pixels per inch? What's going to happen to the size of the image? It's now 100 pixels per inch. How big is it going to be? Still 1 inch by 1 inch? No? No, right. It's going to be tiny now because we have the same amount of pixels, but we're putting them in a smaller space. We have a higher density of pixels, so of course the image is going to get smaller. So if I go back to my image that I showed you guys, the way in which I do this to see the true size of my image at 220 dots per inch is just to open it up in GIMP or whatever image editing program you're using. Go to the image size function, which is on those instruction sheets. You can see right now it's 72 pixels per inch. That's the X and Y resolution. And if I look at how big it is, at this resolution, it's 6 inches by 7 inches. If I want to see how big it is at 220, I have to first turn off any of these interpolations in your instruction. We'll talk about it in 1 second. And then change this to 220. And just like we saw in the example, we have 6 inches by 7 inches. But when that activates, we now see the size of our image at the resolution we want, which is 2 inches by 2.3 inches, right? So that's the actual size we have of the high-quality figure. So what happens next? Now you're in a situation where your image may be the right size, it may be too big, or it may be too small, right? That's the size. That's its honest size for the resolution we need. So how do we know how to change the size to get it what we want it to be in our picture? What proportion of the figure it ultimately will be? And to do that, we just think very quickly about the dimensions of our canvas, which hopefully for you guys is PowerPoint. PowerPoint is normally in landscape mode, but you can simply change it to portrait mode by following these options on your sheet. And the dimensions of the canvas, by default, are 10 inches by 7.5 inches. Okay? So what I would recommend is leaving yourself a 1 inch border, meaning a 1/2 inch border on all sides. So just imagine that's there, and don't put any information in that 1/2 inch border. Just keep it clean, right? You don't have to be too exact, but keep it clean. That way, whenever you embed this in your Word document, which is 8.5 by 11 inches, you'll have a 1 inch margin to be sure you have enough space before you start getting into the information in your picture. If you need more margin than that, just do the calculation to see how much space you need to use, but that's kind of a simple way to do it. So you'll have the added margin 1/2 inch and 1/2 inch. Okay? So now you can start thinking, well, if I have a figure legend, that's going to take up maybe 2 inches at the bottom. The whole slide is this, 9 by 6.5. Half a slide is 4 by 5 by 3.25. So you'll know, like, do you want your image to take up the whole amount of space? Do you want it to take up half the slide so you have an idea of how big it needs to be? And you know how big it actually is. There's 2 things that you can run into. 1, your image is too big. 2, your image is too small. The better problem to have is your image is too big, right? Because then what you do is you throw away pixels. You do that to make the file smaller. It doesn't have to be any bigger than that, which just ensures your thesis isn't some gigantic file size. So when it's too big, you have an image that's 10 pixels by 10 pixels, 1 inch. You can make it smaller, so 1/2 inch by 1/2 inch, just by throwing away the pixels you don't need. And it still has sufficient resolution. You do that very simply. The simplest way is to drag it or insert it into PowerPoint, pick the corner of the image, shrink it to the size that you want, and then click compress. Okay? That's in your instructions. PowerPoint does all the work for you. The second case is more difficult. The second case is when your image is smaller than you want it to be in your figure. Okay? So this we just think about briefly. It's the last problem we have to solve. Your image is too small. So now we're going to think about this. Our actual pixel is 10 pixels per inch, 1 inch by 1 inch, so 100 pixels, the same image we started with. But now we want the image to be the same resolution, 10 pixels per inch, but 2 inches by 2 inches. How are we going to do that? We have 100 pixels, but if we want the same resolution, 10 pixels per inch, now 2 inches, so that's 2 times 10, 20 times 20, but we only have 100 pixels, what are we going to do? What's the only way we can make that happen? Huh? Some mumblings out there. Someone's got something to say. Shout it out. You have to make more pixels, okay? Probably you thought that was too dumb to say out loud, but that's the only answer. Otherwise, you're going to reduce your resolution by half. If you want to keep the resolution, you have to add in more pixels. And how are you going to do that? You can't retake the image. That moment is gone. And the pixel fairy is not going to come in and drop pixels on your image, right? That can't happen. So what a computer can do is it can add pixels for you. And it can do this in a good way and in a bad way. The good way is when it maintains the information in your image, but just adds in more pixels so that it prints out well. The dangerous way is called interpolation, where it actually guesses and puts an intermediate value pixel in between your original pixels to kind of make it look like there's more resolution there than was originally. So when you're showing scientific images, you do not want to interpolate your images when you blow them up, even though you may want to blow them up. And I'm just going to show an example. So when you're enlarging microscope pictures, don't add pixels. If you do, this is what you get. We're going to take this little bitty protrusion here on this synapse and blow it up. You can see that this is good. This is the real information where it shows you the value of those pixels. But it's at 220 pixels per inch. It's just every pixel in this square has the same value. So it made it possible for you to show what it would look like if you actually zoomed up on it so that the reader can see what's in there. Instead of doing this, this is interpolating, where it's adding pixels to try and make it look like this has more information than it does. And people see this who work in imaging, and they know you've interpolated. It looks blurred, and now we don't know what the limit of your resolution was because you've kind of fudged it a bit. There may be some cases where you have really pixelated text, and you want to use interpolation to kind of make it look better. I have tried that, and I have never gotten it to work. You can try it too. The instructions are in your sheet, and it's very simple, but I doubt it will work. And really, to do this is the last demo. It's really simple. If you understand what you're doing, it's absolutely trivial. So here's our original image. We're going to crop this. The instructions for doing that are in your sheet, so that this is now all that's left. We blow it up, and we see the good information, right? We see the pixels that are actually there. We see the information at the resolution. We captured it. This is what we have. This is what we want to show. So the way in which we put this to 220 without manipulating it is just very simple. The same way we shrank the image, we use the same function. And now we keep it at 220 dots per inch. Just make sure that this is turned to none so that there's no interpolation. And now we can make it, put this at inches so it's easier to see. We can blow it up as big as we want. And when it does that, it's going to add in pixels, but it's not going to change the way the image looks. So I'm just going to zoom out. You see it looks exactly the same, right? We still see the pixels that were originally there, but now when it prints out, it's going to look just like this. There's tons and tons of pixels in here, but their only job is to recreate that original pixel unit. Okay? Okay. And like I said, if you guys have questions on that stuff, let me know. But that's the major ways to maintain your resolution, to make sure your images don't look bad. Now you know how to combine them with these other objects that are vectors to ensure that you don't have to worry about them getting rasterized. And then you just save this as a PDF, because PDFs can save both information, and you put it into your document, and the instructions for this are in that handout. Questions? Is everyone feeling comfortable enough that they would actually try this? Okay, good. How many of you guys work with images? A good chunk. Okay. And like I said, I know this was not a lot of back and forth. It's a big group. But when you have questions, just let me know. Okay? Next, I'm going to pass it over to Tom, who's going to show you more, mostly about vector objects, in this case, Excel graphs, and how to use those effectively.
All right, so the plan is I'll go through a quick presentation on making graphs. I'll keep it as quick as I can. And then we're going to give you time to work on whatever you want to work on. If you want to resize some images, we'll all be here to help you do that. If you want to make some graphs, or if you want to draw some drawings in PowerPoint. So the idea is that we're kind of giving this presentation and then giving you an opportunity to practice what we've just gone over.
Oh, Tom, you need the mic.
Thank you. All right, so my booming voice. This is a figure from a senior thesis. Actually, it's from a CEL paper. But we get figures that look like this all the time prior to when we started having this workshop. So what's wrong with this figure? What's wrong with the graph? Tons of things wrong with it, right? What do you think? Text is too small. What else? Colors are too similar. Pastel is for Easter eggs, not for graphs. What else? Gradients are inconsistent. There's no error bars. One of the biggest things that we hear from the readers is that students don't quantify their data. So whenever you're doing something in your thesis, think about a little thing on your shoulder saying quantify your data, and ask your postdoc and your mentors if there's any way you can quantify your data. Okay. And quantifying data means you should have error bars. All right. Anything else? All right. So that's all well and fine. The problem is this is Excel's default. This is what Excel gives you. You need to take control of Excel. Excel is not a great program for making graphs, but most of us have it. Some labs will have much better graph making software that you'll have access to. I encourage you to use it. But if you only have Excel, do not despair. We can work with it. All right. So this is the image. This is the graph pretty much as it appeared in the paper that I pulled this from. What do we think about this? It's better, right? The text is larger. This has to do with colocalization of some stain, and so the colors make sense. There's something labeled red and something labeled green, and red and green make yellow. Anybody have any critiques of this particular figure? There's error bars. All the stuff you guys mentioned is here. Any critiques? Okay, so here's a biology question. How many of you have only one X chromosome? All right. So for those of you who have one X chromosome, there's a non-zero probability that those colors didn't work for you, right? And because boys, males, are often red-green colorblind. So what I would recommend is that you think about that, because there's a non-zero probability that one of your thesis readers, or maybe both of them, will only have one X chromosome. So if you follow this link, this tinyurl.com/colorblindfigures, I made that, it will take you to a blog about color blindness, and that will take you to a link that shows what data looks like to colorblind individuals. It's remarkable. There's lots of different kinds of color blindness. And when you just think maybe like, well, the reader will sort of be able to see it, it just won't look as good to them, it's not true. They won't be able to see it because they're colorblind. It means they can't see those colors. So you should keep that in mind and think about that when you make your figures. All right, so I am going to propose that we take Excel's default graph and we make something that looks like that from it. Because that's still a pretty good graph, right? It's pretty small on my screen, but all of the elements of large text, and it's pretty easy to interpret, are there. OK, so one of the great things about Microsoft is that every single version of PowerPoint, Excel, Word is slightly different so that you can't really compare it to what your neighbor's doing. Google is your friend. one of the things that you should figure out is exactly what version of PowerPoint, Word, Excel you're using. So you go to the About menu or however you do it on your computer, figure out what it is, write it down somewhere so that when you Google, how do I combine images in PowerPoint 2011 for Mac student version 3? Like there's all of these different things and they can matter. Okay? So figure out what version of the software you're using so that you can use Google effectively. All right, so here's the data that were used for this figure. And you can access these data if you want using the tiny URL onFlab4. I don't know how it came up with that, but onFlab4. So you can work along with me now, or when I'm done talking, you can try to recreate these graphs with your own computer and do this. All right, so let us start by making a chart. On my computer, I select the data, and then I go to Charts, and I want this to be, there's all these different options, and I want a clustered column, and there is my awful, hideous color scheme. Okay, so when you're at this point in your thesis, you're probably thinking about how to lay out your figures, and this is a great thing to be thinking about. How do you want your graphs to look? Do you want line graphs? Do you want bar graphs? If you want bar graphs, you want them to be clustered. If you want them to be clustered, how do you want them to be clustered? All of that stuff, if you're drawing that out in a storyboard thing where we're having these storyboarding workshops, then when you actually go to make your figures, you have a sense of what's going on. All right, first thing is that it has chosen the wrong thing to graph against the wrong thing. So the axes are reversed. So we can change that pretty simply. There's a thing called Edit Data Series. Mine is called Select. I can go in here and I can switch the row and the column. And now it's actually plotting. It's switched how it's plotting it. OK. It's also plotting the error that I had calculated. And so we want to get rid of those. So we simply select them and remove them as series. We're getting somewhere here, right? Okay. So something else that I find really annoying is that Excel has decided that even though I don't have any data points above 100, it's going to make the y-axis go up to 120. I find that pretty irritating. So I'm just going to double-click on the axis and fix that now, because it really bugs me. And then as soon as I change that, it's going to make the major unit 10. And I don't like that either. So I'm going to make it 20. And there we go. And I can double-click it again. And we'll talk a little bit about fonts. Right. This is just how my day goes. All right, so fonts. You should think about fonts for your thesis. I'll show you. How many people here know the difference between a serif and a sans serif font? OK, so most people. So I find a serif font, so like Times New Roman, to be a great font for reading written text. I personally, and many graphic image people, prefer a sans serif font for the images. So I like to just pick a font like Arial or Helvetica. I like Arial, but whatever. That is a sans serif font, and that's what I use for all of my images. And then I pick a font like Times New Roman for the text. You're laughing.
No, Helvetica.
Helvetica is a great font. They actually have an entire documentary about the text font Helvetica. You can get it on iTunes, and it sounds bizarre, but it's actually a really interesting thing. Helvetica is everywhere. Anyway, OK. So I like it 14 point. And you know, there are people who think that you shouldn't bold things unless you really want it to stick out. I think that on graphs like this, I really like how the bold looks. So I'm going to make it bold, but that's a stylistic choice that I'm making, and I expect people will disagree with it. OK. Cool. So now to change the color of the graph, what you can do is you can just double click on the series, and you can go to Fill. And you can choose a solid, and let's make this one black. Isn't that nice? And then we can make this one white. And then, of course, we have to go to line, and the default is not to have a line, and so we want a black line, and we want to make sure that it's thick enough that we can actually see it. That's pretty good. OK, I'll leave that one blue for now, but you see what I'm doing, right? Error bars. Different computers handle error bars differently. But let's just put some error bars on here. So I'm going to click here. There's a whole menu called error bars. I like it going up and down. I like the cap. And I've actually measured the error experimentally. So I have my custom error values here, and I can specify, what is this? This is small bud. This is pull to foci. So the error for that is right here. Oops. Doop. And that's the positive value. And then here is the negative value. Uh-oh. What did I do? And voila, I have error bars. And of course, I can click on the bar itself, and I can go to line, and I can make it like bolder so that we can actually see it. OK. So couple of more things that I want to fix. Actually, I don't want to show you the most exciting thing right yet. So here, I'm going to change this font to my Arial 14 point. Of course, it botched everything up, but that's OK. And I'm going to move this out of the way just because it irritates me. So that the graph takes up the whole box. This is much easier to do with a 3-button mouse, but my 3-button mouse broke yesterday. OK. So if I didn't make it quite that big, it's not doing it right now. OK. Well, let's not worry about that too much. So one of the things that I really like about this graph here is that, see how it's like got the small bud on one line and the n on a second line. But here in my cell, like I don't know how to separate those. So there's a way. You have to Google it for your computer. How many people have an Apple on their computer? And how many people have another brand like Sun or Linux or maybe PC? OK. So it's different for every computer. For my computer, I would put where I want the in-cell line break. That's what it's called. I press Control, Alt, Return. And then, check that out. Oops. It put it on a different line. But of course, I can't show you that because I can't zoom this thing down. Come on. It's not a good day. There we go. OK? So now you know how to do an in-cell line break, and that will change how the thing looks on your graph. So now for the most exciting part of this entire presentation. You don't want to go through this for every one of your graphs, right? But once you make a graph that you really like, you'd like to apply that format to all the new graphs you make. This has a high rate of failure, but I'm going to try it anyway. So I select a graph. And I choose copy. And then I select my Microsoft graph. And I can do paste special, formats. So once you make a graph that you like, yes, everybody should clap for that because that, okay. So once you come up with a format you like, you can simply copy the graph and then paste the formats. And it did something kind of cool, like even this, this legend here that was previously didn't have a fill, it put a white fill on it so that you don't see the lines through the legend. So it's actually pretty powerful. It's not going to be perfect, and you might have to still make some adjustments, but it'll save you a bunch of time in clicking every. OK, great. OK, so I'm going to switch. We see, here's a Michaelis-Menten curve. We often see graphs that look like this. This is Excel's default, and it's awful. That's not really, like that squiggly line that Excel puts in is not really data. What you want to do is you want to fit the curve, and you want to show the equation of the curve usually with an r² value. So there's a way to do that in Excel. So you do curve fitting, and I can show people that if they would like to see that. sometimes we'll see like a binding curve. So this would be like a cooperative binding curve. So you'd get this sigmoidal shaped thing, but Excel draws that silly line there, and that looks terrible. So what I would recommend you do, it's not easy to fit that as a curve. You actually have to use a more powerful statistics package to do that. What I would recommend you do is to simply leave it as the dots on the graph, as the plot. So if you're going to do a binding curve like that, unless your lab has access to the kind of software you need to actually fit a cooperative binding curve. Okay? So, Excel, it's not a super powerful program for graphing, but you can get pretty decent looking publication quality graphs if you just learn to use your computer well and harness Google and don't use Microsoft's defaults. So the final thing I want to show you relates back to something Cheryl mentioned earlier, and that is cutting and pasting between things. Okay, so here's my graph. I like it. I'm going to copy it. It's on the clipboard now. And then I'm going to go to Word. And if I just press paste, what happens is awful. So what just happened? What do you see? So the font changed. Like Word decided that its font choice was better than mine. Okay. So let's look at another thing that's going to happen. And now let's say that's too big. So I'm going to resize it. It feels hopeless, and like you might want to. Okay, so okay, ready. I'm going to do File, Edit, Paste Special. And there's all these different options. So one option would be to paste it as a Microsoft Excel chart object. The benefit of doing this is that if I then change the data in my Excel sheet and all goes well, it should update it in my Word document. I would not rely on that. Okay? But something else happened. We lost resolution. I don't know if you guys can see it, but it looks terrible. So what I would recommend that you do, and Word and PowerPoint are the same in this regard, and Ryan will voice up if he disagrees with this, but I would paste it as a PDF. Right? It's beautiful, and you can resize it. And I always hold the Shift key down when I resize, by the way. Do you guys know about the Shift key trick? It forces, so if you resize something and don't hold down the Shift key, it can skew the X and the Y. And so you can get a graph. It makes it look like this, which is what you might see in lecture slides for somebody who prepared lecture slides late at night and wasn't like careful. So just always hold the Shift key down when you, okay. Right, so no resolution loss. My fonts stay the same. Everything looks really beautiful. I can blow it up a lot, and it still looks beautiful. Okay, so my tricks. Make the graph look how you want. Don't accept the defaults. You can always paste the format to get graphs to look similar. I would recommend that you paste them into PowerPoint or Word as PDFs so that you can then manipulate them. I'm done. So I think that you guys should take some time to either draw some vector stuff in PowerPoint. If you have some data that you want to graph, I'm happy to help you with that. Or if you have questions about image and image handling, we're all happy to help you. So thank you for your attention. And we're going to stick around at least until 6. And so you're welcome to work on stuff if you want. So thank you. Do you guys have anything you want to add? One thing that came up today, that I forgot to mention, there's been lots of questions about margins on the thesis. And we will all send our respective concentrators an email about this. We used to have to do this 1.5-inch margin on the left thing so we could bind it. But they've updated the way they bind the theses now so that you don't actually need the 1.5-inch left margin. So we think that the rule is going to be, we want all of the margins to be between 1 inch and 1.5 inches. You choose. I think it makes sense to simply make it 1 inch all around. It looks great. But we'll send a detailed email about that in the coming weeks. So don't fret about margins and fonts.